Hello, everyone. I'm your host, Avery Zimmerman, and today's session is the second installment of our rare disease webinar, Rare Disease Research in the United States, Understanding FDA Guidance and Flexibility in the Application of Regulatory Standards, presented by MMS. As an innovative data-focused CRO, MMS brings insights from a global team of highly respected data and regulatory experts across 4 continents. As a reminder, your microphones will be muted for this session and we'll send out a recording following the webinar. If you have questions, please ask them at any time in the Q&A icon at the top right of your screen. We'll answer them at the conclusion of today's session. Once again, here to present today is Dr. Amanda Viester, Senior Manager of Global Regulatory Affairs, and Ben Casper, Senior Manager of Regulatory Strategy. With that, I'll turn it back over to our presenters.
Thanks, Avery. I want to continue this discussion of the flexible application of regulatory standards and to continue to focus on clinical development. Specifically, I will discuss the points to consider when selecting clinical trial endpoints, considerations for determining the clinical basis for safety and effectiveness of the drug, and finally summarize some of the benefits and uses of natural history during drug development, natural history data during drug development. Next slide, please. So as we mentioned earlier, well-characterized assessment tools or standardized efficacy endpoints may not be available for rare diseases. Because there are many factors to consider, some of which may not even be known in an understudied population, defining appropriate study endpoints can present an intimidating hurdle for drug developers. Discussion with the agency on acceptability of these assessments and endpoints will be required, but sponsors should consider these points when selecting or defining trial endpoints. The FDA's first recommendation for endpoint selection is to take a close look at any evidence of efficacy from early phase exploratory trials. Along these lines, seamless trial designs are especially encouraged, as this makes the best use of every subject that can be enrolled. If we advance the slide, we'll see that the second consideration is a clinical outcome versus a surrogate endpoint. So typically, current clinical outcomes are required for a traditional or full approval. However, use of a surrogate endpoint may allow for accelerated approval, which would typically require a confirmatory study for traditional full approval. However, there are some cases where the pathophysiology of the disease itself prevents a feasible development program, or at least one that would be designed to assess a clinical outcome based on the relatively small population, and so a biochemical outcome may then be acceptable as a full basis, as the basis for a full approval. If we can advance to the next consideration, when treatment assignment blinding is not possible, which may be the case due to the ethical considerations of including a placebo arm or due to disease-related factors that prevent blinding, sponsors should consider the susceptibility of their endpoints to individual interpretation. Objective endpoints are preferred, but are not always possible. Regardless of the appropriate endpoint, effort should be made to minimize bias across the board. For example, sponsors might utilize blinded assessors for performance outcomes and independent endpoint raters that are not involved in any other aspect of the trial. Next slide, please. Next, if a novel clinical outcome assessment must be developed, every attempt should be made to adapt or modify an assessment measure that already exists in that disease, or a surrogate endpoint for the disease under study, if there are any. While selection of clinically meaningful endpoints is preferred, these are frequently subjective in nature, as I was already talking about in the last consideration. While surrogate endpoints require demonstration of analytical and clinical validation, which can be time-consuming and rate-limiting. Thus, consideration of the appropriate endpoint should be one of the earliest initiatives in any new rare disease program. If use of a novel endpoint is likely to be required, this should also be the subject of some of the sponsors' earliest discussion with the agency, especially as natural history data, which can be collected during non-clinical testing is likely to be required for any subjective measure of treatment effect. The last consideration is to evaluate the potential for an endpoint to be used across the full range of patients to be studied. This is especially important for rare diseases because of the size of the population, the practical consideration may be to include a broader range of the disease stages or maybe even genotypes or phenotypes being studied. So I'd like to go into a case study that demonstrates the power and importance of endpoint selection and the FDA's willingness to use their scientific judgment wherever possible, rather than setting a standard that is so high that it's unattainable in the context of a rare disease population. So let's start out by talking about the therapeutic context. Primary hyperoxaluria type 1, or PH1, is a rare and serious disease caused by an inherited deficiency. The deficiency is in a liver enzyme called alanine glycoxylate aminotransferase, or AGT. Early in this disease, excess oxalate is cleared by the kidneys, but it does complex with calcium in the urine to form kidney stones and to form deposits in the kidney, which eventually, over time, leads to a loss of kidney function. As that kidney function declines, the oxalate continues to accumulate and begins to deposit throughout the body and other tissues and can lead to systemic manifestations, including arrhythmias, cardiac arrest, gangrene, bone and joint pain, fractures, and blindness. The role of oxalate in the pathophysiology and progression of the disease was of primary importance, and that's why I wanted to highlight it here in this clinical therapeutic context summary. Calcium oxalate stones, which is the major driver of the pathology of this disease, are slow to form and slow to pass. Similarly, the resultant loss of kidney function, as I already mentioned, it's progressive, but it's generally gradual. Given the rarity of the disease, the FDA determined that it would be challenging to detect the effects on the actual clinical endpoints, this actual decline of the kidney, over the course of a feasible development program. During the discussion of the trial design, the agency agreed that if the change was substantial, meaning that patients with a high baseline level of urinary oxalate would near normalization, that they would accept this as the basis for a full approval. In addition to the rarity of the disease, this agreement was predicated on the knowledge of the pathophysiology of the disease and the role that urinary oxalate plays in disease progression. They also had epidemiological and observational data showing an association between the elevated urinary oxalate and loss of kidney function, as well as between reductions in urinary oxalate and preservation of kidney function. Together with a substantial treatment effect, these data will end up providing the support for the use of this endpoint in this population. Just to wrap up therapeutic context, at the time of this application, there were a few interventions that were available to address the buildup of calcium oxalate and the crystallization that forms. And while these were useful in some patients, many patients still progress. Liver transplant does replace the missing enzyme, which leads to disease resolution, but as you can imagine, it comes at a very high cost to the patient's health and welfare. The actual drug is a synthetic double-stranded small interfering RNA, or an siRNA, and it inhibits the messenger RNA of the hydroxyacid oxidase 1 gene, which is the deficient gene in this disease. Okay, so let's look at the regulatory status. As we've seen in most of the case studies so far, Oxaluma had orphan drug designation, they had breakthrough therapy, and they did qualify for priority review. The data they submitted at the time of the application came from 2 clinical trials. One was a double-blind placebo-controlled trial. It randomized 39 patients that were 6 years of age and older. In this study, they demonstrated a large treatment effect, and they showed a substantial decrease in urinary oxalate from baseline to month 6. Urinary oxalate excretion actually normalized in about half of the patients that were treated. Notably, there was no treatment effect on kidney stone events or kidney function, but as we previously noted, this would not have been expected in a trial that was only 6 months in duration. In the second trial, patients were enrolled that were under 6 years old, the youngest of whom was only 4 months old. This was a single-arm trial without a concurrent control. However, what they found was there was a similar reduction in urinary oxalate, and there was a time course that was generally consistent with the finding in the first controlled trial. So this leads us to the question, what's the outcome? What did the FDA decide, and how is it tied to the rarity of this particular disease? So oxazoluma was approved for treatment of primary hyperoxaluria type 1. FDA partially attributed the success of this program to the input they received from patients and caregivers at an earlier patient advocacy meeting. The application did result in a priority review voucher, and there were no further studies required. This is especially notable as clinical endpoints like kidney stones or kidney function showed no treatment effect. This is a great example of how FDA considers the feasibility of the requirement and uses scientific justification to determine how to apply it. Also note that FDA determined that the data from the second study supported the extension of the indication down to birth. And they noted during the NDA review that they were considering the experience and perspectives that had been shared by patients and caregivers at an externally led patient-focused drug development meeting hosted by the Oxalosis and Hypoxaluria Foundation on October 5th, 2020, which was near the time of the approval. So if we go to the key takeaway, what this case study is intended to show is how essential endpoint selection is, and that the FDA really is willing to set an attainable standard in the context of a rare disease population. Typically, we don't think of surrogate endpoints leading to a full approval, but FDA understood that in such a rare disease population, doing a long-term, long drawn-out study that could actually evaluate kidney function was just not feasible. And they used all of the data showing that there was a direct link between the accumulation of oxalate to support a full approval in this particular instance. Okay, so if we can go on to the next slide. We're still going to be talking about flexible application of regulatory standards in the clinical setting, but here we're going to talk about the overall evidence for safety and effectiveness. There is a statutory requirement for the FDA to base its approvals on substantial evidence of the drug's effectiveness for its intended use and the safety of the drug under the prescribed conditions of use. In the guidance that we've been talking about in this section of the presentation, FDA outlines some of the factors it uses to describe how to interpret and apply this to rare disease research. If we can move to the first of those, it is a requirement. There's a requirement for adequate and well-controlled investigations. This is per regulation. They are required to distinguish the effect of a drug from other influences, such as spontaneous change in disease course, placebo effect, or bias observation. It's important to note that even a single-arm trial can be considered well-controlled, which is what we will discuss in a minute. But first, I want to mention that the design features of an adequate and well-controlled trial protocol are also defined by regulation. These go beyond the considerations for just a control and should be routinely considered in any clinical study design. Okay, so let's talk about historical controls or external controls. Although concurrent controls are much preferred and a randomized controlled trial will be required whenever it's feasible, a historical control may be allowed in certain rare circumstances where therapeutic context makes other trial designs impractical or unethical. In order to allow this, FDA will want to see a well-documented, highly predictable disease course with some components that can be objectively measured and verified. In addition, the treatment effect has to be readily distinguishable, meaning that it needs to be pronounced, compelling, and it should occur in a relatively the same time frame as the intervention itself. So next we'll talk about early randomization. Typically, the agency encourages sponsors to consider randomized controlled trials, despite their difficulty in some cases. They recommend innovative trial designs to allow for randomization even in these earliest phases of clinical development to ensure that each patient's contribution is interpretable. Some activities they suggest to help overcome the hurdles of enrollment include reaching out to patient and caregivers, to stakeholder groups, clinical investigators, and others who can provide contribution in the earliest planning stages, and to propose modified trial designs and have ongoing discussions with parents and treating physicians to address the issues that they may have over the use of controlled arms. Finally, we will talk about the feasibility and the sufficiency of safety assessments. So regulations don't specify the needed evidence of safety, except that the evidence must include adequate tests by all methods reasonably applicable. The FDA interprets this in their guidance on pre-market risk assessment. To the agency, safety in the context of drug development means that the benefits of the drug outweigh its risk for the indicated use or uses. In the serious and rare disease setting, they will apply scientific and regulatory judgment and take particular challenges posed by each drug and disease, including the patient's tolerance for risk in the setting of an unmet medical need, into account when determining what is feasible and what will comprise a sufficient safety assessment. Of note, feasibility judgments do heavily rely on disease prevalence estimates, so sponsors should be prepared to provide adequate scientific justification for those when submitting a marketing application. Also, because of the limited population size in rare disease research, FDA may encourage the use of auxiliary or other supportive safety cohorts. These can be populations with a related indication. These can be the matched controls that we've been talking about. There's many different formats that a supportive safety cohort may follow. If we move to the next slide, we'll talk about natural history studies. Natural history studies are covered extensively in FDA guidances, both in the common issues guidance that we've been talking about for several slides now, but also in a separate specific guidance on this type of study, which can be interpreted in the context of rare disease. There is one thing to note. When FDA refers to a natural history of a disease, they're using the traditional understanding of natural history to mean the course a disease takes in the absence of intervention. Conversely, when they talk about a natural history study, they understand that patients may be receiving the current standard of care and possibly even emergent care. And they actually list these differences of treatment modalities as one of the variables that natural history studies should identify. Although the FDA does not require natural history studies, a certain amount of natural history of the disease is necessary, absolutely necessary, to inform drug development. Where that doesn't exist, a natural history study is likely to be required. So on this next slide, we will talk about the benefits and uses of natural history. So we've already discussed where limited knowledge of the disease is a hindrance to orphan drug development. The data from a well-designed natural history study can help address some of those gaps in the understanding of the disease. The areas of specific importance to drug development are listed on this slide. Natural history studies can help define the disease, identify the patient population. They can lead to appropriate selection of endpoints and early recognition of safety concerns. Overall, they can help identify the different critical elements of a clinical trial design. Also, natural history data collection can benefit the whole rare disease population in ways that are not directly tied to the development of the single drug under development. FDA encourages making natural history study data publicly available to support and promote rare disease drug development for this reason. Next slide, please. For this case study, I want to take a look at a program where data from subjects in a natural history study were actually used as an external historical control. So let's start out by looking at the therapeutic context. The disease in this case is two similar diseases, Hutchinson-Gilford progeria syndrome and progeroid laminopathies. These are both rare and serious genetic diseases. They cause premature aging and death and have a debilitating effect on people's lives. These patients experience accelerated cardiovascular disease from a buildup of the defective proteins in the cells of the heart. Tragically, many patients died before the age of 15 years from heart failure, heart attack, or stroke. At the time of this application, there is only supportive care and symptom management available. So the active ingredient in Zokinvy is a farnesyltransferase inhibitor, which was first in class at the time of this application. I'm going to talk about safety and efficacy studies in a minute, but first I wanted to talk about something else that was submitted along with this application, and that was the mechanistic studies that were done by the sponsor. These provided important, objective, confirmatory evidence for both indications. So what they were able to show is that lonafarnib, or Zokinvy, prevented farnesylation and then the subsequent accumulation of the farnesylated progerin in the inner nuclear membrane of the cells. And by showing this predicted mechanism of action, they were able to strengthen their application. Not only the predicted mechanism of action, but it was a mechanism of action that directly addressed the canonical pathophysiology pathway of this disease. Similarly, they used mechanistic studies to bridge the data from the less rare Hutchinson-Gilford progeria to the more rare processing deficient progeroid laminopathies. So as you'll see in a minute, I can't separate these out for the next slide, but as you'll see in a minute, only one patient was included for this very, very rare disease application. But the mechanistic studies showed that fibroblasts from both types of patients responded similarly to the drug, and that scientific justification was there to show that the two diseases shared a similar pathophysiology, mechanism of action, and disease manifestations. Okay, so moving into what was submitted for the safety and efficacy of this drug. So two small single-arm clinical studies enrolled patients, and they overcame the limitations of relying on a non-randomized study by using well-matched external natural history study data. In addition, during review, there was numerous issues that were identified. These included the potential for type 1 error and bias. Both of these were overcome, in part due to the objective mortality endpoint that was selected. So I know that was a previous point that we've discussed, was the importance of the endpoint, but it was both in this case, the well-matched natural history data, as well as the objective mortality endpoint that are very important to understand for the ultimate decision that was made. So what was shown was that there was a survival benefit in the HGPS patients that was established by comparing mortality. The follow-up time was 11 years for the single-arm clinical studies and 13 years for the unmatched treated cohort. At the time of this submission, the lifespan was increased by about 3 months in the first 3 years of this follow-up, and by about 2.5 years through the maximum follow-up time of 11 years compared to untreated patients. So given this information, what was the outcome and how was it tied to the rarity of this disease? Well, the FDA decided that a mortality benefit for this serious, devastating disease that didn't have any other treatment options overrode any of the risks identified in the application. As I mentioned, there was risks identified throughout. This was both in the non-clinical and clinical programs, and some of these will be addressed in the post-marketing commitments that the FDA has required. However, the FDA felt that the identified safety concerns did not outweigh the benefit of an improved mortality, and that remaining investigations could be conducted post-approval because this treatment was so desperately needed by this patient population. So the key takeaway here is that relying on natural history study data as an external control may be acceptable in cases where it's not feasible or ethical to use an internal control, particularly in rare disease populations. So if we move to our next slide, we're going to shift our focus away from rare diseases as a whole to human gene therapy products in the treatment of rare diseases. This specific subset of therapeutics for rare diseases are those that seek to modify or manipulate the expression of a gene or to alter the biological properties of a living cell for therapeutic use. These products are biologic. They mediate their effects by transcription or translation of a transferred genetic material, or by specifically altering human host genetic sequences. Some example of gene therapy products include nucleic acids such as plasmids, in vitro transcribed RNA, and others, genetically modified organisms such as viruses, bacteria, and fungi, engineered site-specific nucleases, which can be used for human genome editing, and ex vivo genetically modified human cells. Development of this type of therapeutic for rare disease is covered in its own guidance, which will cover manufacturing, preclinical, and clinical trial design issues for all phases of clinical development. Much like the common issues in rare disease guidance that has been the earlier focus, this guidance identifies elements that are not specific to rare diseases, but that are recommended when investigating a gene therapy product and are likely to be issues for rare disease populations. FDA intended this guidance to assist sponsors in designing clinical development programs for these products where there's a limited study population size, as well as where there's potential feasibility and safety issues relating to the interpretability of the data. And some of these may be unique to rare diseases or may just be due to the nature of the gene therapy product itself. So first, I'd like to briefly cover the CMC considerations. So note that FDA clearly states that the CMC considerations for product manufacturing, testing, and release are the same for human gene therapy products, regardless of whether they're intended to treat a rare disease or not. They do, however, acknowledge some of the challenges due to product development strategies that are brought about by the limited population size. And the limited population size, one of the considerations there is that it's going to take a reduced number of lots and fewer times manufacturing the therapy, and this can shorten the amount of time that sponsors have to set up the critical process parameters and identify the critical quality attributes. So FDA has noted the importance of well-controlled manufacturing processes and suitable analytical assays to assess product critical quality attributes for product concentration, potency, identity, and purity as early in development as possible, optimally before clinical use. They also encourage use of innovative strategies for understanding these CQAs or critical quality attributes. And they note that this may include applying prior knowledge from other similar products, leveraging product characterization data from non-clinical studies, and evaluating the critical process parameters during engineering runs, or the production of multiple small lots versus a single large production lot, which would allow identification of some of these parameters and attributes during development. Sponsors developing gene therapy products for rare diseases are strongly encouraged to contact the FDA prior to the IND submission and throughout drug development to discuss product-specific characterization and considerations for CMC. A preclinical program that is tailored to the investigational product and the planned early phase clinical trial contributes to the characterization of the product's benefit risk profile for the intended patient population, whether that is in gene therapy or as Ben was talking about in the overall drug development. The overall objectives for a non-clinical program or preclinical program in gene therapy are listed here on the slide. And a lot of these are very similar to what you would see in a standard or typical non-gene therapy drug development program. However, the preclinical program for any investigational product should be individualized with respect to scope, complexity, and overall design. And the FDA will accept proposals with justification for any potential alternative approaches, such as in vitro or in silico testing. However, these should be submitted early during early communications and meetings with the FDA. So moving on to clinical considerations, there are several considerations for clinical trials, and I'm only going to touch on a few of them today. They generally fall into the categories I've listed here on the screen. If you take a look at these, you might be thinking, well, aren't these the same considerations as for clinical trials in other products? And you'd be right. But under each of these in the guidance, the FDA has listed out specific considerations that pertain directly to the use of gene therapy or to the diseases that gene therapies are typically used to treat. For example, when thinking about study population, if the disease is caused by a genetic defect, FDA recommends that the sponsor must perform genetic tests for the specific defect of interest in all clinical trial subjects. And because of the potential for phenotypic heterogeneities, there is a wide variance in the onset and severity of disease manifestations. Therefore, subjects with severe or advanced disease might experience confounding adverse events that are unrelated to the related to the underlying disease rather than to the GT product itself. So it can be difficult to detect a benefit from therapy in those subjects. So FDA recommends using subjects with earlier stages of the disease that might derive more benefit from the therapy. For dose selection, another recommendation the FDA makes is that sponsors have to consider transgene expression. That's obviously specific to human gene therapy. There's also considerations of innate and adaptive immune responses when considering safety considerations. So for the sake of time, I'm not going to go through examples for each of these, but the guidance does lay out numerous examples for most of the categories that I've shown here. Okay, so the end of this guidance briefly touches on the RMAT, or Regenerative Medicine Advanced Therapy designation, as well as other expedited programs. I won't talk about those too much today, as that's part of the next webinar, but there is a key takeaway interwoven in that discussion, and that's that the FDA recommends early and frequent discussion before submission of the IND, but also calls out that even before that, even before you're starting to think about your earliest interaction, there may be an earlier point where you can meet with the FDA, and that's through CBER's initial targeted engagement for regulatory advice on CBER products, or INTERACT meetings. These meetings are used to help sponsors talk about development issues for preclinical development and manufacturing before they're even ready to get into an IND. Because of time, I don't want to do a full case study on a human gene therapy right now, but I wanted to at least mention one landmark application. Luxturna was the first directly administered gene therapy approved in the US. It was the first, that targeted a disease caused by a mutation in a specific gene. It was actually the third overall. The first two were gene therapies for oncology indications, but this was the first one to address a rare disease that was caused by a mutation in a specific gene. It was approved in 2017, so the same year that the earlier two were approved, and it treated a specific form of retinal dystrophy that led to vision loss and sometimes to complete blindness. There are numerous summaries available of this landmark approval, but the stories of restored sight are truly remarkable, and they reinforce the potential of this fairly recent medical breakthrough of human gene therapy. And how it can be used in the context of rare disease drug development. So with that, I will pass it on to Ben to talk to us about ultra-rare disease research.
Thanks, Amanda. So far, we have discussed case studies, regulatory guidance, and precedent for rare and ultra-rare indications. But what about the rarest of the rare diseases? Those impacting such a small number of patients that on their own, they are unlikely to be commercially viable, even with current FDA incentives? The short answer is that we really don't know yet. This is a relatively untested area where there is limited guidance and precedent to explore. However, through the increased connectivity of the modern world and the dedicated efforts of patients, their families, physicians, and researchers, along with the FDA's willingness to modify the existing framework and accommodate these extremely critical and time-sensitive development programs, we are seeing the first steps of a path beginning to form. So before I touch on some precedent, I would like to discuss two fairly recent FDA guidances. The first and most recent guidance is the development of individualized antisense oligonucleotide drug products developed to treat severely debilitating or life-threatening diseases that are caused by unique genetic variations where only a small number of individuals are prospectively identified. Or as the guidance states, usually one or two. This guidance is particularly exciting as it is directed specifically to those doing research on those very rare indications and outlines basically, here's what you need to do or get done in order to get into the clinic. The guidance itself states that it is not intended to address non-clinical testing for commercial development of oligonucleotides and ends by clarifying that expansion to a larger population or an intent to commercialize a treatment would typically warrant additional studies. However, as research into very rare diseases is often parent-driven, this guidance, which outlines abbreviated non-clinical assessment approach, may be their bridge to researchers that are willing to work with them, especially given the low likelihood that such treatment would ever be licensed. So the guidance appears to have been well received by the ultra-rare disease communities and has been applauded by those creating individual treatments for patients with ultra-rare diseases caused by genetic mutations. The second guidance lays out clear recommendations for grouping patients with different molecular alterations for eligibility in clinical trials and outlines preferred approaches for studying low-frequency molecular alterations. This allows for evaluation of subgroups of a clinically defined disease defined by the specific molecular alteration or group of molecular alterations that cause them, even when occurring in only a few subjects. I'll discuss this more in a minute, but as in many things, we are finding that with rare disease research, there is strength in numbers. Guidances like these allow us to group individuals with ultra-rare, even N of 1 mutations that can increase the likelihood that a therapy will be made commercially available. However, this guidance does not cover individualized therapeutics, like the AOS guidance. Rather, this is discussing a situation where a single target therapy may be effective in multiple groups of patients who have different underlying molecular alterations because the functional effect of the molecular alteration may be similar. The guidance outlines the development and regulatory considerations for this class of therapeutics. So just a few items to note. In this context, the FDA encourages broad indication language to encompass those that would have been eligible for clinical trials, irrespective of whether or not they were actually represented. Secondly, although the agency does not numerically define a low-frequency molecular alteration, they state that these are occurring at frequency low enough that enrolling a sufficient number of patients to conduct a clinical trial limited to the specific molecular alteration of interest is not feasible or practical. Finally, other types of evidence in combination with or independent of clinical studies may also support a grouping strategy for clinical trials. In addition to clinical evidence demonstrating the proposed group of specific molecular alterations exhibit similar responses to the investigational drug, the FDA may accept non-clinical, in silico, or mechanism-based evidence. In certain cases, extrapolation of evidence from other drugs in the same class could also provide support for a proposed grouping strategy, as could the identification of certain consistent disease-related phenotypes. So with that, I will go into another case study. So as I mentioned earlier, there's limited regulatory precedent for clinical research of an ultra-rare disease identified in only a few patients. This is in part due to the fact that the populations in this whole category of disease are too small to attract industry effort. And as of now, there are still no commercial incentives to directly address these diseases. However, we can learn from what is being done in the academic and nonprofit sector. And this is an example of that. So this case study is for Malaysin, which is an investigational drug for a rare form of Batten's disease identified in a single patient. So in 2018, it was precedent setting in that it was the first time that the FDA had allowed for use of the expanded access investigational new drug pathway to allow for research of a new drug created for a single patient. Prior to this, expanded access had been limited to allowing for use of existing drugs in seriously ill patients with no other options. To overcome some of the regulatory challenges, this program built on the success of a development program for a similar drug with the same platform. Both Milicin and Nusinersen are antisense oligonucleotides. They are similarly sized, they have identical chemical modifications, and they both target CNS tissue. Nusinersen, or Spinraza, was another rare disease success story and was approved in 2016 for the treatment of another rare CNS disease, spinal muscular atrophy. Given this information, what was the outcome and how was it tied to the rarity of the disease? Well, in January of 2018, the FDA did grant approval for expanded access for the treatment of the rare form of Batten disease caused in part by the insertion of the retrotransposon CLN7 gene. So in this case, although there was no existing FDA guidance and no regulatory precedent, the time from diagnosis to synthesis and treatment was still less than a year, around 12 months. So in a world where next generation sequencing is revolutionizing the diagnosis of genetic diseases, this case certainly demonstrates a nascent pathway for the individualized treatment of ultra-rare indications. So key takeaway is that this is an example of individualized genomic medicine. A novel therapeutic agent for a patient with a rare disease was able to be treated through an expanded access program. So individualized therapeutics, also called bespoke therapeutics, have become increasingly feasible due to scientific advancements, allowing us to detect and understand the molecular underpinnings of these diseases, and as well as the simultaneous technological advancements that allow for development of a toolbox to help address needs out of reach from traditional commercial drug programs and not likely to ever be licensed. So currently this toolbox includes things like gene therapies based on the use of specific vectors such as adeno-associated viral vector to deliver a specifically engineered transgene insert that may vary between patients. Or genome silencing or editing technologies, genetically engineered bacteriophages, or custom formulated bacteriophage cocktails for multi-drug resistant infections, and manufacturing of next generation sequencing based on therapeutic products for the treatment of cancer. As mentioned previously, the FDA is starting to put guidance in place for the development of individual therapies like these. However, there are a few other resources that I wanted to mention, and these include a public workshop put together by CBER in March of last year, the transcript and webcasts of which are available online and contain a lot of helpful insight into the individualized therapeutic space. As I already mentioned, there are strength in numbers, and developing a standardized approach to regulate and license individualized therapies may contribute to their advancement. A few efforts I wanted to call out. First, at the NIH, platform vector gene therapy researchers are using a common gene delivery vehicle, an adeno-associated virus, or AAV, to create gene therapies for 4 rare genetic diseases currently being studied at the NIH Clinical Center. In this many diseases at a time approach, the NIH team is working to improve the efficiency of the clinical trial startup process for gene therapy and develop a standardized blueprint for similar gene therapy products focused on rare diseases. Finally, I want to acknowledge the tireless research of those at the NIH and at various research hospitals around the globe, as well as contributions from the foundations like the N. Lorem Charitable Foundation and numerous other rare disease and/or patient advocacy foundations. So the key takeaway for this entire talk, as we have mentioned in numerous times throughout the talk, it is extremely important to get agency buy-in on your plans, whether for CMC, non-clinical, or clinical development. Talk to them about what is needed to support clinical trials, talk about the overall non-clinical program, and talk to them early and often throughout the drug development process. Time and time again, the agency, and in particular the OOPD, has demonstrated their motivation and seemed to be quick to engage and open to scientific justification wherever possible. Scientific judgment is subjective, so while precedents and guidance help, it's impossible to know the FDA's opinion without asking it. Also, do not assume that what applied to another rare disease program will automatically apply to yours, and get the necessary designations to help ensure a successful clinical development program and approval process. And with that, I'll end the talk, and thanks everybody for your time today.
Thank you, Amanda and Ben, for your insights. Now we'll be moving forward with the Q&A portion of the webinar. I'll hand it back to the presenters.
Thanks, Avery. I will read the first question. I saw the press release pertaining to MMS providing pro bono support to ultra-rare disease patient advocacy groups. How do we apply? Ben, do you want to take this one?
Sure, so it's pretty straightforward. You just need to send an email to the attention of the Ultra Rare Disease Assistance Program, and that's at media@mmsholdings.com. So that's media@mmsholdings.com. And you can request an application through there. And just so that everybody knows, the first review of applications will occur in mid-October of 2021.
Okay, and the second question, you mentioned that natural history studies can also benefit the rare disease population outside of just drug development. Can you elaborate? I'll take this one. The FDA does give several examples in their guidance on this topic. Some of these are just that these type of studies provide a nuclear center for evaluation of this disease. So of a particular disease that can be facilitating communication, creating or even finding disease-specific centers of excellence, as well as other benefits to having a place to go when you find out you have this disease. Another benefit that's not directly tied to drug development, but is certainly related, would be that that these studies can help create a standard of care, or if there is a standard of care, they could even potentially identify improvements. And I think one other thing is that, and this is why FDA asks that you make your data publicly available, and that's that there's data now on the disease that did not exist before. This can be demographic or other data on disease characteristics. It could also be things like prevalence or disease tracking. All of these things can be helpful to a wide range of development programs and not simply tied to one single product's development program. So that is it for questions. So I will turn it back over to Avery.
If you have any additional questions on what you heard today, please follow up with your point of contact directly or email media@mmsholdings.com and we'll direct you to a presenter. The next edition of our rare disease series will take place on October 6th and will focus on orphan drug, rare pediatric disease, and expedited program designations, FDA incentives to promote rare disease drug development. To register, visit mmsholdings.com/webinars or follow us on LinkedIn for updates. Thank you very much for joining us today.